Hi folks, my name is Cole, and today on Investigate, Explore, Discover, we're going to be looking at how antimicrobial resistance is increased at wastewater treatment plants. Now, I think at this point, most people have directly benefited from using antimicrobials. For example, if you've ever gone to the doctors with an earache or upset tummy, they've probably prescribed you antibiotics to clear up the symptoms. Now, antibiotics work against bacteria, but we also have chemicals that work against viruses, parasites, and fungi. And together, these are termed antimicrobial agents. Antibiotics, in particular, are pretty much the cornerstone of modern medicine since the 20th century. So we've used them to gain the upper hand against microbes when dealing with infections. However, bacteria have developed ways of getting around these antibiotics. And if antibiotic treatments are not adhered to or they are used in too low concentrations, this can allow some bacteria to survive which are resistant to those antibiotics and become the dominant population, which means that when somebody else is infected with those, the antibiotics will be ineffective in clearing that infection. Bacteria can react to different antibiotics by changing different things inside of them, such as reducing the amount of drug in the cell. They can destroy the drug, they can change the drug targets, or they can develop methods of making the process that the drug targets redundant. Now, these mutations can arise spontaneously or can be transferred from other bacteria on mobile genetic elements called plasmids. Bacteria can do this relatively quickly, with the advent of antimicrobial resistance occurring just a few years after antibiotics commonly become prescribed. Antimicrobial resistance can occur because it can spread so easily between bacteria in humans and wildlife, but particularly in water systems, where concentrations of antibiotics can be relatively low, but just enough to provide positive selection pressure to influence these bacteria to become resistant. To summarize this, though, antimicrobial resistance can spread between humans, between animals, and through the environment. And this gives bacteria a way to fight back against our tools against them. So antimicrobial resistance is a huge problem that already exists in the world, one that the World Health Organization has identified. Now, from a study in 2014, it was estimated that there were over 700,000 antimicrobial resistant or AMR related deaths. But if nothing is done to augment how we use antibiotics, this number could change to over 10 million deaths worldwide by 2050. And this is not projected to affect the world evenly either, with most AMR related deaths projected to happen in Asia and Africa. This problem is not without economic costs as well. If we don't do anything about antimicrobial resistance, the global cost of it by 2050 could reach over $100 trillion. But there are steps that we can take to mitigate this. There is a proposition that $2 billion per year be spent on a global innovation fund for early stage research on antimicrobial resistance. And there is already agency collaboration between the World Health Organization, the Food and Agriculture Organization, and the World Organization for Animal Health to combat antimicrobial resistance. But to deal with antimicrobial resistance, it really requires a multifaceted approach, which should include other agencies such as law enforcement, policymakers, those involved in agriculture, in communities, and even pet owners, to coordinate, communicate, and collaborate together to set up antimicrobial resistant surveillance networks to prevent infection and control them, to initiate antimicrobial stewardship, and to fund research and innovation for new antimicrobial compounds. Together, this will help achieve the best health outcomes for people, animals, plants, and the environment. If you take away nothing else from what I'm saying, have this message burned into your mind. As a species, we need to stop consuming antibiotics like they're candy, because otherwise we won't be able to use them anymore. Now, the World Health Organization has already published a list of problematic bacteria and labeled them according to their particular threat in hospitals, nursing homes, and among patients whose care requires devices, such as ventilators and blood catheters. The bacteria that we're looking at today is a priority 1 bacteria, and that bacteria is Escherichia coli, or E. coli. Now, these bacteria are normally present in our healthy gut flora. However, pathogenic strains are a leading cause of bloodstream and urinary tract infections. This gram-negative rod-shaped bacteria has a few strains that can cause severe disease, such as diarrhea, vomiting, stomach pains, and cramps. However, most people do recover in about a week without the need for antibiotics, but some people don't, and they do need them. Infection with these types of E. coli usually happen by eating contaminated food. Now, E. coli is a very diverse species, and we've come up with ways to classify them. We've classified them according to their genomic content and labeled this phylotyping. So these E. coli can be grouped into different phylogroups or clades. E. coli are also indicators of fecal contamination of water environments. Of particular note is that there are numerous studies that report a considerable presence of AMR E. coli in waste environments. And it has been previously found that contamination of surface water has been due to clinically important strains. Now, something important to ask of all science really is, why is it important? So why is it important to look at E. coli antimicrobial resistance levels in wastewater plants? Now, it's important because previous to the study that we are looking at today, there has been no systematic whole genome-based study which is focused on the difference in the prevalence and characteristics of AMR E. coli strains between sewage and river water. This gives us clues as to figure out the roots of AMR transmission and to identify bacterial population trends. Now, the name of the paper that we are looking at today is "The level of antimicrobial resistance of sewage isolates is higher than that of river isolates in different E. coli lineages," by Ogura et al. From the Kurume University School of Medicine in Kurume, Fukuoka, Japan. And they investigate antimicrobial resistant E. coli in different water environments. Particularly, they look at southern Japan in the Miyazaki Prefecture. So they look at 2 river sites, one of them upstream and surrounded by forest in the Keita River, and one of them downstream of an urbanized area in the Kiyotake River, and compare these to different wastewater treatment plants. When collecting samples from each of these sites, they identified the bacteria and found that over 75% of the bacteria identified were E. coli. And to further classify them, they separated these E. coli into their phylogroups and found that these phylogroups are spread throughout the collection sites. Of particular note, though, is that phylogroup F was rarely seen in sewage sites. So next, they looked at antimicrobial resistance in samples and found that just under a fifth of them were resistant to antibiotics. They found in river settings the rates of AMR was very low. River settings also had the highest rates of clade 5 organisms, which are susceptible to all antibiotics. Sewage plants, E. coli strains had more antibiotic-resistant phenotypes and genes, and there was a higher prevalence of multiple antibiotic resistances. So when investigating these antibiotic-resistant genes, they found that 73% of them were of known gene origin, so they've been documented before. However, 27% of them are of unknown gene origin, so they just spontaneously arose. This is some pretty interesting information and significantly contributes to identifying AMR levels in different water environments. Particularly, this study gives examples of how prevalent AMR is in sewage treatment plants compared to rivers, which shows the human impact on AMR and sets a benchmark for identifying patterns throughout the world. I think that all science can be used as a stepping stone for more science in the future. And I had a few thoughts on some interesting directions to take this research moving forward. So firstly, these samples were collected in August to November, And there's previous evidence suggesting that microbial populations change in a specific site depending on the season. So how could antimicrobial resistance rates change across seasons or bacterial populations change? Furthermore, because they only looked at sites in southern Japan, how does this compare to the rest of Japan, or around the rest of the world for that matter? And finally, to do something about this, I thought it would be interesting if there were ways that sewage plants can implement practices to reduce antimicrobial resistance. These are just a few of the thoughts that I thought about when reading this article. But what I would like to know is what sort of ideas or questions popped into your head when hearing about this information. So let me know about that in the comment section below. Ultimately though, I hope that you learned something, and more importantly, that you enjoyed your time doing so. So if you did, give this video a like and subscribe for more content in the future. With that, I will see you next time.